everyone. My name is Pete Strutton. I'm a lecturer here in IMAS at UTAS. So I just want to, at the beginning, explain, if everyone's not clear on this, is that we have Centre of Excellence people who come in from universities all over Australia participating this week, but we also have about 20 or so students who are actually taking this class for credit. So we, as part of the Quantitative Marine Science program, which is a joint program between UTAS and CSIRO next door, we have a core set of classes that QMS students take, and so there's about 20 students that are taking this as QMS 512. So if you haven't already, those students, if you haven't already been there, this is the Milo site, and this is where you can get all the material for the class. And I just want to point out that I'm not the unit coordinator. Xana Chase is the unit coordinator over there. So if you have any questions about the logistics of the class for the QMS students, just talk to Xana. I should probably say something about just logistics. So we're in the IMAS building here. So this building was completed a couple of years ago. It was part of basically stimulus money from the government in 2009. So it's really nice we used to be on main campus, now we're down here right next to CSIRO, which is excellent. The bathrooms are just out there, sort of past where you registered, and also the computer lab where we'll have the lab session this afternoon is there. And I should probably say something about emergencies. If there's a fire alarm, we should all just scream and panic. And then we just go outside the front door and sort of towards the temporary fencing over there. There's a muster area around there. There'll be someone with a hard hat on. Okay. Okay, are there any questions right now before we get started? Okay, so this class is marine. It's originally marine biogeochemistry, but this year, when we're running it as part of the Centre of Excellence, we're also going to include biogeochemistry of the terrestrial biosphere and the atmosphere as well. But we're going to start out talking about biogeochemistry of the oceans, and there's Xana's already put online links to a textbook that we'll be following partially for this first part of the class. That's Sarmiento and Gruber, Ocean Biogeochemical Dynamics is the name of the book. And I'm going to start out today talking about nutrient cycling and productivity in the oceans. So in the oceans, so we should maybe start out by saying that the oceans are responsible for about half of the photosynthesis that happens on Earth. So if you want to think about it in terms of oxygen production or carbon cycling, about half of the oxygen that's produced or that is in Earth's atmosphere is from photosynthesis that occurs on land, and about half is from the oceans. And in the oceans, that's mostly carried out by single-celled plankton, phytoplankton. There are, of course, other photosynthetic organisms in the ocean like seaweeds and kelps, but as a proportion of total oceanic photosynthesis, they represent an extremely small part. So about half of the oxygen in the atmosphere comes from the oceans, and about half of it comes from the terrestrial biosphere. So I often say that if you want to gain an appreciation for how important phytoplankton are, you just skip every second breath. And in the ocean, we have phytoplankton at the surface of the ocean. They need to be there to carry out photosynthesis because that's where the sunlight is. They're consuming nutrients, and they're basically taking dissolved nutrients out of the surface of the ocean, packaging them into their cells, which are then consumed by higher trophic levels, and some of those cells sink out. And as that biological material sinks out, it gets remineralized back into the dissolved fraction. So when I say remineralized, I mean that these biological particles are raining out of the surface of the ocean, and they're being attacked by things like bacteria. Bacteria are consuming oxygen to basically redissolve that particulate material back into the dissolved phase. So if we want to think of this in terms of an element like nitrogen, in the surface of the ocean, we have nitrogen that's there would be nutrients here. Nitrogen that's dissolved mostly as nitrate, NO3. Phytoplankton use sunlight energy to perform photosynthesis and package that nitrogen up into particulate organic nitrogen. then those phytoplankton are either consumed by zooplankton or they rain out when they die, and then bacterial activity remineralizes that particulate material back into dissolved carbon and nutrients. So there's sort of I like to think of it as a sort of a competition between biology and physics. Phytoplankton require sunlight, and they also require dissolved nutrients in the surface of the ocean. So wherever we get active transport of these dissolved nutrients back into the surface waters, we're going to get potentially high productivity. And so areas where this transport is very active might include places like coastal upwelling systems. So think of coastal California or Peru. So actively transporting nutrients into the surface waters where phytoplankton can combine that nutrient source with light to produce organic matter. So it's that process, that sort of competition between physical processes and biological processes, that sets the global distribution of ocean productivity. So phytoplankton are photosynthetic organisms, so they contain chlorophyll. So one way that we can map the productivity of the oceans is by looking at the surface distribution of chlorophyll. So chlorophyll we can use as a proxy for productivity. It's an imperfect proxy because high chlorophyll levels don't always necessarily mean that high productivity is occurring. But this is a map here of global distribution of surface chlorophyll concentration. So this is just a representation of a satellite image. Here's another way of looking at this. This is a global biosphere, so it's chlorophyll concentration both on land and in the ocean. So we see some large-scale patterns. We see very high productivity in places like these coastal upwelling systems, so the coast of Peru, California and Oregon, the Benguela and the Canary upwelling systems off the coast of Africa. You'll note that the west coast of Australia is unusual in that it doesn't have a coastal upwelling system. All the other continents do on their western coasts. The reason that we don't have upwelling on the west coast of Australia is because of the Lewin current, which is flowing in the opposite direction for what usually generates upwelling. So this is a map of global chlorophyll, and we can see, as well as those productive upwelling systems, we can see this is an average over the entire life cycle of this one satellite, CWIS, that measured chlorophyll. And in the wintertime, productivity in the Southern Ocean is extremely low, but if we average it over the year, it's actually quite high. And we see other productive regions in the sort of the lee of South America here. And we also see there's a seasonally very high productive high productivity region in the North Atlantic, and I'll talk a specific example about that later on. Okay, so this high productivity in the surface waters is being fueled by nutrients. What do we mean by nutrients? Anna's going to talk about elemental cycling in the ocean a bit more than I will. But we can define a nutrient as any element that has the potential to limit photosynthesis. So phytoplankton are taking up nutrients to perform photosynthesis. The main nutrients that they're using are probably nitrogen, phosphorus, and for some phytoplankton, silicon. They're also taking up carbon and water. The concentration of carbon in the ocean is relatively high compared to all of those other elements that I just mentioned. So the concentration of carbon is never limiting to the productivity of the ocean. So what we call the macronutrients, so elements like nitrogen, phosphorus and silicon, they're present at concentrations of the order of, say, 1 to 30 millimoles per metre cubed. Note that millimoles per metre cubed is also the same as micromolar. There are other elements that we refer to as micronutrients, and a good example would be iron, that I'm going to talk about in detail later. And that's present at concentrations that are about 1,000 times lower than the macronutrients. And there are large areas of the ocean where the concentration of iron is so low that it does limit productivity, and I'll talk about that in detail. So carbon and water are basically never limiting to photosynthesis. We can represent the photosynthesis equation like this, or if you look at the textbooks, Sarmiento and Gruber, they write it out like this. So we're basically taking carbon, nitrogen and phosphorus combined with water to make, we could call this fixed organic carbon, particulate organic carbon, carbohydrates, whatever you like to call it. So it's basically the phytoplankton are taking these dissolved nutrients and packaging them into particulate organic carbon, so making cell parts out of dissolved nutrients. So what we're going to do in the next couple of slides is look at the distribution of nutrients with and without the biological pump. And I should, so I sort of use interchangeably, I'll use terms like productivity or biomass or photosynthesis or the biological pump to mean this process here of photosynthesis. We call it the biological pump because you can think of this biological productivity taking CO2 out of the atmosphere into surface waters, packaging it into particulate organic material that then rains out into the deep ocean, or you're pumping carbon from the surface waters into the deep ocean. Okay, so this is a map of the surface concentration of nitrate in the oceans, compiled from thousands, probably millions of measurements. So I said that the macronutrients like nitrogen in the ocean vary in concentration from from 0 to about 30 micromolar or millimoles per meter cubed. So so this is the surface distribution of these nutrients. What I should probably say, going back to the very first slide, if we were to look at the concentration of nutrients in as a function of depth in the ocean, we have quite low concentrations of nutrients in much of the ocean near the surface. And then as this biological material is raining out, it's redissolving back into these dissolved nutrients. And so if we were to plot a profile of nitrate, for instance, in the surface ocean, it would be low at the surface and it would increase as we go down in depth. So typical concentrations might be of the order of 1 to 5 millimoles per meter cubed at the surface, going down to about 30 millimoles per meter cubed for nitrate in the deep ocean. So, but we do see concentrations of about 30 millimoles per meter cubed of nitrate in some parts of the surface ocean. And this is due to limitation by iron. So anywhere where we see nutrients that are hanging around at the surface, there must be something that's preventing the phytoplankton from taking up those nutrients. And in the Southern Ocean, the tropical Pacific, and the North Pacific, it happens to be the concentration of iron that is limiting the uptake of these other nutrients. Now, one of the reasons that we get high nutrient concentrations, I'll just go through a couple of examples, like why do we have high nutrients in the Southern Ocean, why do we have high nutrients in the tropical Pacific? I'll just explain the physics behind that. So this is Antarctica. So what I'm showing here is basically we're looking at a cross-section down in the Southern Ocean here. So in the Southern Ocean we have winds, very strong winds that blow from west to east. So if this were plotted as a globe, we'd have winds sort of encircling Antarctica. And because of the rotation of the Earth, Coriolis, when the winds blow in the southern hemisphere from west to east, we get a transport to the left, a transport at the surface of the water to the left of those winds. So we've got here in the Southern Ocean, we've got winds blowing from west to east. So there's a strong transport that's set up in the same direction as those winds, but there's also a transport that goes to the left in the southern hemisphere, and the northern hemisphere would be to the right. And that generates coastal upwelling. So the winds are blowing, water's being moved away from the Antarctic continent. Water has to come up from below to replace that water. And as I just said, the water that's at depth has relatively high nutrient concentrations. So those high nutrients are brought to the surface, and they can potentially fuel that high productivity that we saw in the satellite image. We saw high chlorophyll around the coast of Antarctica. Interestingly, because of the very low iron concentrations in the Southern Ocean, a lot of that nitrate and phosphorus and silicon is not used by photosynthesis. And when those cold waters get to the subtropical front, they're subducted into, here it says AAIW, Antarctic Intermediate Water. We also refer to these as mode waters. And that water gets subducted and over the course of decades, it's eventually going to be upwelled in other parts of the ocean, including the tropical Pacific. And that's just illustrated on this figure here. So here we've got the upwelling. So it's calling this circumpolar deep water upwelling. Some productivity occurs at the surface here, but it's not reaching its maximum potential because of limitation by iron. And then relatively high concentrations of nitrogen and lower concentrations of silicon are subducted into these mode waters. So that says subantarctic mode water there. And then those mode waters then go into the lower latitude ocean and even into the North Pacific and North Atlantic. And it's thought that those subducted nutrients can fuel up to about 70% of productivity in the rest of the ocean. So what happens here is incredibly important for what nutrients are left over for the rest of the ocean to use. Okay, so just going back to this figure here, I've just explained the origin of this high nitrate water around Antarctica. Let's talk a little bit about what's causing the relatively high nutrient concentrations on the equator there. So this is a map of the tropical Pacific. So here's the Americas, here's Australia and Papua New Guinea. So under normal conditions, there's trade winds that blow from the eastern Pacific towards the west. Those trade winds do two things. First of all, they generate upwelling along the equator. So the trade winds are blowing right along the equator. In the northern hemisphere, the Coriolis transport, or the Ekman transport generated by Coriolis is to the right, in the southern or to the north. In the southern hemisphere, it's to the left or to the south. So there's divergence. I think the next figure probably shows this a little better. Yeah, okay. So here we are. We're standing on the Galapagos Islands, and we're looking towards the dateline or towards Papua New Guinea. So the winds are blowing in this direction. They're causing equine transport to the right in the northern hemisphere, to the left in the southern hemisphere, and those waters have to be replaced by water coming from below. There's a fatal flaw in this argument, and that is that at the equator, Coriolis is actually zero. So saying that equine transport is generated to the right and to the left is probably technically not true, although I guess as you go a little bit away from the equator, Coriolis starts to increase, but it's good enough for today's story anyway. So those waters are coming up from below, and they cause cooler sea surface temperatures on the equator, and also these winds are sort of piling up this warm water in the Western Pacific. So that's the origin of these relatively high nutrient concentrations on the equator. But the amount of macronutrients like nitrate that are upwelled relative to the concentration of iron, the ratio of nitrate to iron is high, or you could think of it as the amount of iron that's being upwelled is relatively low. So this is another region of the ocean where we have low iron concentrations and we have high nutrient concentrations hanging around at the surface. That water, when it's upwelled, is also bringing with it dissolved inorganic carbon, or dissolved CO2. So this is a plot showing based on several million measurements of surface CO2 concentrations. This shows the flux of CO2 between the ocean and the atmosphere. So anywhere where we have reds and yellows, the ocean is a source of CO2 to the atmosphere, and anywhere where we have purples and blues, the ocean is taking up CO2. So the strong productivity that happens in the North Atlantic serves to suck up CO2 and make that region a sink of CO2. So the tropical Pacific is the largest oceanic source of CO2 to the atmosphere. So it's a massive region. It's about a quarter of the Earth's circumference. And the amount of CO2 that's degassing out of the tropical Pacific is about half of the US fossil fuel emissions every year. So it's a really significant part of the global carbon cycle. Okay, so I've gone through a couple of examples of how we get nutrients actively transported to the surface waters, so the large-scale upwelling that occurs around Antarctica and the upwelling that occurs on the equator. So these are examples of where we're getting those two important resources for photosynthesis. So we're getting nutrients and light co-occurring in the same place. Another way, another phenomenon by which we can get those nutrients and light co-occurring in the same place happens every summer when the ice retreats around Antarctica. The same is also true in the Arctic. The expansion and retreat of ice that happens around Antarctica is probably one of the biggest, physical perturbations to the Earth's system that happens on a seasonal cycle. As the ice forms, it's excluding salt, so that the sea ice that forms around Antarctica is largely frozen fresh water. Also, that ice can accumulate phytoplankton cells by a couple of different mechanisms. So when that ice starts to melt in the summertime, that's fresh water or frozen fresh water that's melting on the surface of the ocean. So it makes a relatively fresh, low-density layer on the top of the ocean. And also, any phytoplankton that are frozen into that ice will be released into that freshwater lens. So this represents a favourable environment for photosynthesis to occur. So what happens when the ice retreats around the poles is that we can get a bloom, a phytoplankton bloom occurring in that freshwater lens. So here's a very high-tech drawing of how that occurs. So this is wintertime here, where we've got the maximum ice extent, and as that ice formed, and also throughout the winter, it accumulates phytoplankton cells on the bottom. As we go into spring, that ice starts to melt, and so it's frozen freshwater, so it makes a freshwater stable lens on the surface of the ocean. It's also releasing some of these phytoplankton into that freshwater lens, and that makes a bloom that's right around the ice edge. As the winds blow during the summertime, they start to break down that freshwater lens, and so that sort of degrades this favourable environment for photosynthesis to occur. So as the ice is retreating, we see this bloom that basically follows the ice edge as it retreats. And you can see that not only in my very artistic diagram, you can also see that in satellite data. So here's average November conditions in the Southern Ocean. So all of the white that we see here is ice. So what I'll do is I'll toggle through the months of the spring and the summer, showing the ice retreat and the bloom that in many places around Antarctica follows the retreat. So here is this is the Ross Sea, and this part of so the reds and yellows are high chlorophyll concentrations, so productive seawater. This is the massive Ross Sea polynya. So polynya is just a area of sea ice that opens up, and this one permits high productivity. But as I toggle through the months, you'll see I'll sort of point out a few places where the ice retreats, and you see high chlorophyll concentrations where the ice retreated. So actually in this example, you see the Weddell Sea polynya starts to open up, and you also see some high chlorophyll around the northern part of the Weddell Sea where that ice has retreated. Okay, and then you can see as we go in, now we're in January, the ice that disappeared around, that's the Bellinghausen and Amundsen Sea, that's generated some high productivity as well. So this mechanism of a stable upper water column that has phytoplankton released into it stimulates high productivity. And during a intensive field study that was conducted in the Ross Sea area in the late 90s, this is a fairly complicated diagram, but basically what it's showing is through a series of cruises, they went down there and sort of followed the retreat of the ice. And as the ice was retreating, there's a bloom occurring at the ice edge, which is depleting. This plot here is silicon concentration. So as the bloom follows the retreat of the ice, it's leading to this strong gradient in silicon and this sort of serial depletion of silicon as the ice retreats. So I'll talk a little bit about phytoplankton diversity in a minute, but silicon is an important element for one particular type of phytoplankton, and those are the diatoms. So phytoplankton are single-celled organisms that contain chlorophyll, and they photosynthesize. They take up inorganic carbon out of the surface waters. the the diatoms, as well as doing all that, they have glass shells around the outside of their cells, and so they make that out of dissolved silicon, and so they are the major players in the surface ocean for the uptake and the cycling of silicon. Okay, so I've just gone through some mechanisms that generate these relatively high nutrient concentrations in the surface waters. So that's this is data here. So this is a map compiled from observations. This is a similar map from a biogeochemical model. So this is a computer simulation that's trying to represent the cycling of elements in the ocean. And so I guess what I'd like to say first about this is that I think it does a fairly good job of simulating the global surface distribution of nitrate. The interesting thing about, I think you're all aware that the part of the reason that we do computer modelling is to try and understand the system, but we can also do experiments that we can't do in the ocean. For instance, turning biological productivity on or off. So this is a biogeochemical model simulating the surface distribution of nitrate. What happens if we turn off the biological cycling of nutrients? So in a global ocean, after 5 years, if we have no productivity, so at the very beginning I talked about how biological productivity is working to deplete nutrients at the surface. As that biological material rains out, it gets remineralized back into the dissolved fraction. So it's biological productivity that is causing these low nitrate concentrations in places like the North Pacific and the South Pacific subtropical gyres. So if we turn off biology for 5 years, this is the distribution of nitrate in the surface waters. And I think this is kind of surprising, actually, that after 5 years with no biological productivity, the distribution doesn't look that much different. We haven't homogenized the ocean. What this tells me is that there is some spreading of these high nitrate areas. So it seems like lateral mixing, or lateral diffusion is mixing some of these high nitrate concentrations from the surface areas where it's high into the lower areas. But this suggests to me that vertical mixing in the ocean, so if we've switched off biology, we've switched off that process that's sucking up nitrate in the surface waters and delivering it to the deep ocean, vertical mixing is really doing almost nothing to redistribute that high nitrate concentrations from depth to the surface. So I think this is a really interesting representation of how important biology is in the distribution of elements in the ocean, and how important or how not important vertical mixing is. And if we switch off biology for several thousand years, then nitrate concentrations in the global ocean get completely homogenized. So lateral mixing and vertical mixing have worked to almost completely homogenize nitrate concentrations in the global ocean. So now nitrate basically looks a lot like salinity. Okay, so we're going to break in a little while, but what I'm going to talk about now is the processes that are controlling productivity in the ocean. So when I talk about productivity, it's basically the synthesis and the export of organic matter out of the surface ocean into the deep ocean. And I want to, in this part of the lecture, I want to understand, or help you to understand, what controls the recycling efficiency in surface waters. So by recycling efficiency, I mean the cycling of that particulate organic matter back into the dissolved fraction, where it can be reused by photosynthesis. Okay, so I've just talked about vertical mixing. So I'd like to think about mixing and vertical transport as two different processes. So mixing is the breaking down of those gradients in nutrients in the vertical, whereas vertical velocity, or this plot here, is basically the movement of water masses up and down in the ocean. So what this shows is so positive values mean vertical transport towards the surface, and negative values mean transport away from the surface. So what we see is that those the strong upwelling that occurs around Antarctica is associated with these strong positive velocities. We also have positive velocities in the tropical Pacific. I talked about the mechanism that's generating upwelling there. And in the subtropical gyres, so poster child here probably being the North Pacific, we have negative vertical velocities, so those gyres are net downwelling. So if you were to look at the circulation in these gyres, in the North Pacific here, the circulation is in a clockwise direction, which is piling up water in the middle of the gyre here and sort of pushing down the waters in the centre of the gyre. So that's why we have a net negative vertical transport, and that's what leads to those places being very low in productivity. There's very weak vertical mixing bringing nutrients to the surface, and there's a net transport of water if we don't think about mixing, if we think about vertical transport, it's going down in the middle of those gyres. So it leads to those places being very low productivity regions. Another process that's really important for setting the limit of productivity is the depth of the mixed layer. And I'll talk about this in the context of the seasonal cycle in detail, but this shows the global distribution of the winter mixed layer depth, and we see extremely high or extremely deep winter mixed layers in the Southern Ocean. This is caused by the very strong winds in the Southern Ocean, and this shows you an example or a plot of that. So especially in the Southern Hemisphere winter, we have very strong winds and very deep mixed layers in the Southern Ocean. So this is important for regulating ocean productivity for two reasons. First of all, when you have extremely deep mixed layers, any phytoplankton that are present in the surface ocean are going to be mixed down. So you think about that mixed layer overturning on the time scale of approximately days. So any phytoplankton that are present in the mixed layer are going to be, in this case here in the Southern Ocean, they're going to be transported from the surface to 300 metres depth over the course of a day. And so especially in the wintertime when there's low light levels anyway, they're going to be exposed to, on average, very low light regimes. So it makes it almost impossible for them to photosynthesize. So you can't get high productivity in areas where we have very deep mixed layers, especially in the wintertime. But there's another reason that deep mixed layers are important for productivity. As those, as I mentioned before, nutrient concentrations are low at the surface and high at depth. So as the ocean's being mixed down to depths of 300 or 500 metres in the Southern Ocean, it's eroding down into those very high nutrient concentrations, and so it's homogenizing very high nutrients in the surface waters. So that means that when summer comes along and the mixed layer starts to get shallower and light levels start to increase, the phytoplankton are able to take advantage of those very high nutrient levels that have been mixed into the surface waters through the course of the winter. I think I'll just skip that one. Okay, so I've talked about the processes that can bring nutrients to the surface, so I just wanted to talk a little bit about the interplay between nutrients in the ocean. So this is a plot made from a bunch of measurements from the Atlantic, the Pacific and the Indian Ocean, and it's nitrate concentration versus phosphate concentration. So what this shows is that here we're going from, this might be waters that have been recently mixed to the surface, so they have relatively high nutrient concentrations. As we move down this line, we're moving along a gradient of biological uptake of these nutrients. And you'll notice that the intercept here is not at zero. So when we get down to zero nitrate concentrations, we still have some phosphate that's present in surface waters. And the slope of this line is 16, which is the ratio at which nitrate is taken up relative to phosphate. And this is sometimes called the Redfield ratio. So the ratio by which carbon and nitrogen and phosphorus are taken up by phytoplankton is 106 for carbon to 16 for nitrogen to 1 for phosphorus. So for every mole of phosphate that's taken up by photosynthesis, 16 moles of nitrate are taken up and 106 moles of carbon are taken up. So this says something about what might be the ultimate limiting nutrient of productivity in the oceans, and it suggests that that phosphorus is the ultimate limiting nutrient because it's going to the amount of phosphorus that's left that determines the amount of nitrate that's taken up. So this is the cycle of nitrogen in the ocean. So on the y-axis here, we're basically going through oxidation states of nitrogen, and the most biologically available form of nitrogen are these low redox states here. So the least oxidized or the most reduced form of nitrogen. So phytoplankton take up nitrogen. They prefer to take it up as ammonium if they can, and they can package that directly into particulate organic nitrogen. As that nitrogen gets remineralized back into the dissolved phase, it goes first into a form that's close to ammonium, and then it gets oxidized by the oxygenated seawater back into nitrate. So the most abundant form of nitrogen in the oceans is nitrate, which phytoplankton can take up, but it's energetically very expensive to do so. So they need to expend energy and use enzymes to get it into this organic nitrogen phase. And then it can, if it's remineralized effectively in the mixed layer back into ammonium, it can be taken back up rapidly. And this is what happens in the subtropical oligotrophic gyres. So another important process by which nitrogen can get incorporated into the surface ocean is by what's called nitrogen fixation. So there are certain phytoplankton, or actually they're cyanobacteria, that have enzymes that can directly take dinitrogen, that's 80% of Earth's atmosphere. They can take that nitrogen gas and synthesize it by a process called nitrogen fixation into organic nitrogen, and then that becomes through this process of remineralization, that can become available to other organisms in the surface ocean. So this represents a mechanism by which new nitrogen can get put into the ocean. There is no corresponding process for new phosphorus to be put into the ocean. So that's why it's thought that the cycling of phosphorus sets the ultimate limit of productivity in the oceans. And this is just another representation of that process. So nitrogen fixation that's carried out by diazotrophs, so cyanobacteria, takes this nitrogen out of the atmosphere and then can package it into phytoplankton or biomass, which is then cycled in the surface ocean. So this is meant to represent phytoplankton, zooplankton, and bacteria. So incorporation of this new nitrogen into the food web represents a new input of nitrogen to the ocean cycle. And then places where there's active transport of high nitrogen levels from the deep ocean into the surface ocean are places where you can get strong productivity, and so I explained a few of those, the Southern Ocean, coastal upwelling regions, and the tropical Pacific. And then that biological material can be exported into the deep ocean as dissolved organic nitrogen or particulate organic nitrogen. So this wiggly arrow here is meant to represent export of particulate material and remineralization back into the dissolved phase. Okay, so I think we'll break there for a little while. We'll just take a 5-minute break and we'll come back and start again at the top of the hour. Yeah, so that's what happens in the oligotrophic gyres. So there's not much new input of nitrogen from below, and that there's very small phytoplankton that exist in those parts of the ocean. They're efficiently grazed by microzooplankton, and so that nitrogen is cycled very rapidly in the surface waters. Yeah. Yeah.
Are there secret Wi-Fi details for those that can't get on EduRoam? Annette's on program leave, so we think UNSW's kicked her off for this while she's on maternity leave.
Oh,
And she
okay.
can't get on.
Can't get on the internet, yeah.
Can't get on EduRoam.
No.
And we wonder if we think it's because UNSW's.
Okay, so what about the what's on your? That's just for logging into the
The lab, I think, isn't it?
yeah, I guess so.
it's not, I haven't tried that. But
I don't think that's. I think that's just for the lab.
You know what you could do is I think you could try Uconnect. So if you look
yeah,
at
I went on the Uconnect their setup, and then they're moving towards EduRoam and to use that.
Oh, okay.
So I was like, okay.
Well, what if
Okay.
What if I just
Well, maybe you could use that login on EduRoam.
Okay, I'll try that.
Why don't you try that?
try that? Otherwise, I could just log in.
Yeah.
See if that works.
Very okay.
I haven't used EduRoam actually. If I use wireless in this building, I use Uconnect.
Yeah.
But yeah, you do have to go through. First, you have to connect to Uconnect setup, and you probably have to install something, and and then go through to and once you've done that, you connect to Uconnect, and you could maybe use that login
Yeah,
to
I'll
do
try
that,
that and see if it
or
works.
you could try that on
Yeah.
EduRoam.
It's not like this. Not everyone's.
Oh, Annette's on maternity leave at the moment, so we're thinking UNSW might have kicked her off the system.
Yeah,
Because she's not
It's a
supposed to be
unique
working.
situation. Not supposed to be working.
She's not really here.
I'll try that.
Hey, Chris. Yeah. More nitrate relative to phosphorus. Yeah. I don't really know the reason for that. Yeah. And then the Pacific is falling below the line. It's probably related to the rate at which those two elements are remineralized. And so the Pacific is older water. So yeah, it's potentially, if one is remineralized faster than the other, then that's going to cause changes in that ratio.